Hello, friends, and welcome back to the channel. It's always beautiful having you here as we discuss various and diverse concepts in science. Today, we are going to look at why do we do a PCR. When we do a PCR, we know that we make many copies of a gene of interest, a sequence of interest, make thousands or millions of copies. So the question we're trying to answer today is, why are we making so many copies of this DNA? What is the importance of making so many copies? Number one importance is that the DNA that is usually recovered from any of the samples that we may be using is usually very small in quantity. Very minute amount of DNA is usually recovered when we are trying to do our DNA extraction from the different samples, whether it's a bacteria, a virus, a fungi, whatever it is. We usually are able to recover or extract just very little DNA. And even though we have this small quantity of DNA, our gene of interest is even smaller, because remember that the gene is just a section along the length of the DNA. So it is even smaller, and it makes it difficult to study. So in order for us to be able to study it adequately and to be able to make sense out of what we are studying, we need to make many copies of this DNA or this sequence of interest. We need to make many copies of them to make detection possible. If we don't do so many copies, detection will be quite difficult. Also, there's the issue of analyzation and characterization. Many of the downstream operations that we do, and when we say downstream operations, we are saying that after we have done the extraction of the DNA, and then you do the PCR, your electrophoresis, when it now comes to your actual sequencing, many of these analysis, you need to make use of sufficient amount of DNA or sufficient copies of the gene of interest. And so this is another reason for which we need to make many copies, so that we can have many copies of this sequence of interest in such a way that we are able to study it adequately. Let's say whether it's a sequenced gene, whether also when we want to identify mutations, for an example, to be sure along the length of the sequence where the mutation actually happens, we need to study different copies of the same gene or different copies of the same sequence of genes. We want to study different copies to be able to identify optimally and adequately the exact place where this mutation might be occurring. Also, when we want to look at gene structure and gene function, to be sure of all these things, we need many copies of the same gene. Then many times we want to identify a gene, then also when we do studies on gene expression, you can have a gene and the gene can either be turned on or turned off. And what do we mean by that? You have a situation where the gene is leading to phenotypic characteristics. That is, you're actually seeing that gene, the characteristics of that gene. Or the gene could be turned off, where the gene is present, but in that particular generation, the gene phenotypic characteristics are not being shown from that gene, although that gene is present. So in all these kind of studies where you are doing gene expression studies, it's very important that we make many copies of the gene to help us in our analysis. Also, when we try to identify virulence genes, we try to identify stress response genes, we try to identify antimicrobial genes, it is important that we make many copies of these genes to be able to identify the actual point. Loci is what we call it, the actual point along the length of the gene where these genes or where these sequences are located. This is very important, and it helps us to do crucial work when we are working on antimicrobial resistance, when we are working on pathogenicity of different microorganisms. This is important for us to do. And then when we are doing studies where we are doing cloning, we are doing gene fingerprinting or DNA fingerprinting, all of these kind of studies is very important to make many copies of the gene of interest. We'll do a video where we'll also now continue and now talk or discuss extensively on the different types of PCR. We'll talk about them one by one, conventional PCR, real-time PCR, quantitative PCR, nested PCR, all these different types of PCR. We'll take them, maybe 2 at a time or 3 at a time. Remember to follow us in this journey, scientists. Don't be tired. Remember to like our content, share our content, and do have a blessed day. Cheers.